Hello and welcome to the third part of our lecture on inflammation repair. For this lecture, we'll be talking about the different patterns of acute inflammation, and we'll be having a quick rundown of the chemical mediators that are involved in the inflammatory process. We'll be talking about what the outcome is for acute inflammation. We'll be talking about the different morphologic patterns, and as mentioned, we'll be having a brief run through of the chemical mediators. Now, what happens after everything has been said and done? What happens when after acute inflammation? No, essentially, four things happen. Acute inflammation does what it's supposed to do, and there is complete resolution. There is complete removal of the inciting agent. The necrotic tissue and the dead cells are taken out, and um, there is healing of the tissue, okay, and that is called complete resolution. Sometimes healing occurs, but the connective tissue, but the cells that make up the tissue, the damaged tissue, is not replaced by the original type of cell. It is instead replaced by scar tissue, and this is known as fibrosis. Now, in quite a number of cases, the inciting agent is not removed. In this case, the tissue progresses to chronic inflammation. In some cases, the organism is killed, and um, the dead tissue, and this produces a lot of dead tissue, dead WBCs, but this is not actively removed. The dead material is not removed by the body. As a result, you have a collection of pus or purulent material, and this is known as abscess formation. Okay. Now, as what I have hinted at earlier, um, when you try to stop inflammation, when the inflammation has subsided, a number of processes actually take place. This involves number one, the complete removal of the inciting agent, but equally important is the removal of the main players of acute inflammation. So the WBCs that actively took part in the killing of the cells will have to be removed. The chemical mediators that have been secreted have to be neutralized. Um, the vascular permeability has to be returned to normal, and WBCs need to stop going to the area of inflammation. Also, the cells that have been killed, the microorganisms that have been destroyed, have to be cleared out from the body. Now, there are different acute inflammation may present grossly or may present clinically in different ways. And the types of inflammation will actually tell us the extent or the degree of the inflammatory process. Okay, so we have four morphologic patterns of inflammation. You have serous, fibrinous, suppurative, and ulceration. Serous inflammation is just the outpouring of thin fluid. So this is actually just transudation. Okay, now essentially what happens in serous inflammation is that um, fluid or plasma just leaks out through increased movement of fluid from the blood vessel into the surrounding tissue. So it's the plasma that is just leaking out. So this is known as serous inflammation, and it is it is characterized by the leakage of fluid, and this usually manifests as a blister. Okay? Now, if the degree of endothelial contraction is great enough, what's going to happen is that there is going to be um, movement, not just of fluid, but also of small proteins, particularly fibrin, into the extracellular space, in which case this is known as fibrinous inflammation. Okay? Um, So essentially, what makes it different is that ang degree of endothelial retraction is greater. So it's not just fluid that leaks out, but also fibrin. Okay. Now, the fibrin that goes out can either be resorbed through by by the process of several enzymes, and this is known as fibrinolysis, or there may be growth of connective tissue, and this is known as organization. Okay? So this picture shows us an example of fibrinous pericarditis. Okay? So you will see clots forming in the pericardium. Okay? And you actually have thrombi here. Okay? Or fibrin deposition. And a third type of morphologic pattern is catarrhal inflammation, and you see this when there is outpouring of mucoid material in the mucosal surfaces. For example, a runny nose. Okay, so that's an example of catarrhal inflammation. And the greatest extent of acute inflammation is when you actually have the cells leaking out. Okay, and the WBC. So you will have purulent material, and this is known as suppurative or purulent inflammation. Okay? So an abscess is an enclosed collection of pus. So essentially, this is dead cells, neutrophils, and other necrotic debris that is lined by fibrous tissue that is walled off from the rest of the body. So this is known as an abscess. Okay? So here you will see this one, it's lined by fibrous tissue. So we can see the demarcation here. But inside, you also see dead cells and neutrophils. Now, an ulcer is a local excavation of the surface of an organ. Okay? And so if it is full thickness, as what you can see here, then it is known as an ulceration. But if it is not full thickness, you call this an erosion. Full thickness ulcer. If hindi full thickness, this is known as an erosion. Okay, so here you can see a defect in the epithelial surface, and then you have a base that is necrotic. And then here you have exudates, here itong bright pink na material, and then you have granulation tissue or healing tissue underneath. Now, some terms also have fistula, which is a connection between two surfaces. Okay. So a common example is an anal fistula. So here you can see the anus, the opening of the anus. Frequent infection will facilitate the connection, another connection into the surface, and this is known as an anal fistula. Okay. Now, another term that you need to be aware of is cellulitis. Cellulitis is infection of the interstitial tissue just beneath the skin. So I think you might have experienced this in some form or another. So this is usually inflammation of the underlying tissue just beneath the skin. Okay. Now, what are the chemical mediators of inflammation? So we've actually talked a bit about some of them, but basically these are chemicals that to some extent participate in the inflammatory process. So how are these produced? They are usually produced either through the presence of microbial products, through certain pathways that lead to the production of these chemical mediators, and these would include the complement system, the coagulation system, and the kininogen systems. Okay. Now, as with all things about inflammation, they have the potential to cause harmful effects. Therefore, mechanisms need to be in place to control these reactions. Okay? And the control of these reactions may be brought about by the decay of these substances, gubonia, enzyme inactivation, or actual elimination of the mediators. Like, for example, the glutathione scavenging system, which will actually neutralize reactive oxygen species. Now, chemical mediators may be derived from the plasma, or they can be produced by the cells. And the cell-derived mediators can be produced by different cell types, including the macrophages, mast cells, endothelial cells, and leukocytes. Now, one of the most important mediators that we have encountered are vasoactive amines, and these include histamine and serotonin. They play a part in the vascular response. Okay, so these are the chemicals that are predominantly responsible for the vasodilation seen in inflammation. Okay, so histamine is produced by many cell types, particularly mast cells and basophils, and we have different stimuli for release. So ang most important functions niya is to cause dilation, vascular dilation, and increase vascular permeability. So it is inactivated by histaminase. Another related vasoactive amine is serotonin, and this is produced by platelets. Its action is quite similar to that of histamine. When you have allergies, you have massive activation of mast cells, and they secrete large amounts of histamine. Okay, and this is the reason why when you have allergies, you will have swelling because there is massive movement of fluid out of the blood vessels. You will have redness, you will have itchiness. If the extent of the allergic reaction is so great, then there is massive efflux of plasma out of the blood vessels, and this will rapidly decrease blood pressure. This is known as anaphylaxis or anaphylactic shock, which is fatal. Okay? Have eicosanoids, which are produced from arachidonic acid. They produce prostaglandins and leukotrienes and lipoxygenases. Okay? So we'll be talking about this in pharma na. But one of the most important prostaglandins is thromboxane A2. Thromboxane A2 is responsible for the initial vasoconstriction. Okay? Thromboxane A2 is responsible for the initial vasoconstriction. Generally, prostaglandins are vasodilators. Okay? Prostaglandins are vasodilators. The only exception here is thromboxane A2. Okay? Leukotrienes are Leukotrienes are responsible for spasm of the bronchi and increased vascular permeability. Leukotrienes play a major role in the vasoconstriction associated with asthma. Okay? And lipoxygenases are another group of substances produced by arachidonic acid, and they serve as anti-inflammatory agents. Again, you have three types of eicosanoids. Eicosanoids are chemical mediators that are produced from arachidonic acid. Okay? And these include cyclooxygenases, which includes prostaglandins, lipoxygenases, and leukotrienes. Okay? Also have platelet activating factor. Again, it's a major player in inflammation. It serves to increase vascular permeability and facilitate movement of leukocytes. Okay. You have markers for acute inflammation. The most important would be TNF, interleukin 1, and interleukin 6. The mother cytokine is TNF, or tumor necrosis factor. Okay. Interleukin 1 is primarily responsible for vasodilation, movement of the WBCs across the blood vessels. It is also responsible for systemic effects, the general feeling of being sick. This is brought about by TNF, interleukin 1, and interleukin 6. The most potent cause for fever is actually interleukin 1 acting on the thermoregulatory centers of the brain. That's why you will have fever. Okay? Interleukin 1, TNF, and interleukin 6 can also act on the bone marrow to produce to facilitate the movement of WBCs out of the bone marrow into the blood. Okay? Interleukin 1 and interleukin 6 also act on the liver to produce acute phase reactants, which are opsonins. Okay? Another chemical mediator are interferon gamma and interleukin 12, which are actually markers of chronic inflammation. Okay? Of course, you're aware of reactive oxygen species. They facilitate destruction. And the result is the production of bleach or hypochlorite from oxygen gas. Nitric oxide is a short-lived gas that is also produced during inflammation, and it serves to facilitate vasodilation. Okay? The WBCs also facilitate secretion of enzymes through their lysosomes, and these include the release of proteases. Okay? Neuropeptides are also produced by inflammatory cells, particularly in the lungs and the GIT, and they serve to transmit pain signals and regulate vessel tone. Okay? Also have different types of proteases. The kinin system, which is associated with the production of bradykinins, which plays a role in pain. Okay? The complement system, okay, which facilitates phagocytosis, opsonization, chemotaxis, okay, and cell lysis, and clotting systems, which leads to the formation of blood clots. Okay, so list the most likely mediators. So these tables are actually just a summary of everything. So you can pause the video and list them down. So that's it for chemical mediators. Thank you very much for listening.